All right, welcome to biology. We are now continuing on with our conversation about nucleic acids with our biomolecules, but we're going to be focusing on DNA and really in particular, and I'll make this a little larger. Oh no, that's not what I meant to do. DNA replication and how that then starts the process of protein synthesis. Really looking at our learning target. And now thinking back as I'm talking about this, I should have written learning target in here. One of the connections we need to make is how can the code within our DNA, how can those nucleic acid bases really create, our bodies and our proteins, and again, the proteins are what make up our eye pigment, our eyeballs, our hair, our skin. Every part of us is really a form of protein that we can think of. Other than, of course, the fats and carbohydrates, but really the structures and that make us who we are forms of protein for the most part. And so these proteins have to be created, right? We've talked about, and we probably know from previous science classes, that DNA is the code for life, and it's kind of the blueprint that makes each of us slightly different, right? So we want to talk about and truly start to understand, well, how does that work? Um, and so today, we're focusing on two different things. It's DNA replication, and I wanted to talk about this because it really uh, goes into how the DNA works in the sense that it can copy itself, that it can create new copies of itself continuously, and that it's in every single cell that we have in our body. Every single nucleus contains our DNA, and so it's constantly copying itself. And uh, down the road, we're going to talk about, okay, so it's copying itself over and over and over, and there's billions of these nitrogen bases here in the nucleic acid in our DNA, and so to copy billions of those, millions, if not billions of times, to be in every single cell, we're going to talk about, well, uh, what's the likelihood that mistakes happen? Do mistakes happen? And what are those? So we want to have a general understanding of, how does it make a copy itself? And then we can talk about how does that then form proteins, but then also what happens if a mistake happens? And does that impact the proteins that get formed? So that's kind of where we're headed with this, and the conversations we're looking to have. So, just a little review. We did this right before spring break, but here is our basic DNA structure. Again, we've got our phosphate groups and our ribose, really, right? This is deoxyribose, and then our nitrogen base. So again, this is a carbohydrate sugar, and one of the big takeaways that we focused on last time was that this sugar and phosphate, but sugar and phosphate creates the backbone of our DNA, and without it, uh, our DNA would not exist. So carbohydrates and that sugar is really used in our DNA, and we also talked about how those carbohydrates and sugars are used to make fats as well, and for energy. And so um, carbohydrates are incredibly important, and these sugar molecules really are the foundation to life. And with that, these sugar molecules are mostly carbon. So again, we're called carbon life forms, and that's just a quick recap of what we've covered. But so now looking at this again, we have our carbohydrate and phosphate, and then our nitrogen bases: adenine, thymine, guanine, cytosine, Okay, and we did talk about briefly that adenine must bond with thymine and guanine with cytosine and so forth, cytosine to guanine, however you want to talk about it. They bond together, and I like this image because it really highlights the reason why is that adenine and thymine to be well I would call happy. When I think about chemistry and bonding and what happens naturally, chemicals want to be relaxed. They don't want a lot of tension, and the tension is going to come from uh, different charges, positive and negative charges, and possible binding sites. And so they want to find something that complements what's available in their structure. And so adenine and thymine both want two hydrogen bonds to happen. And so therefore they end up complementing each other, while guanine and cytosine want three hydrogen bondings to occur. And so they end up complementing each other. And so therefore they're going to be attracted to one another, and then they're going to kind of click like a puzzle, right? So uh, again, little recap. We talked about the bonding together. So bonding patterns, I highlighted it here just as an important piece of where we're going. Because as we create the replication, right, and copying of itself, uh, that there's going to be different enzymes that need to know and help with this process and need to put the right nitrogen base with the correct pairing. So to help with this, it is, to be honest, a very complicated process, and so I looked long and hard for a video that really tries to break it down simply. So this video, I want to say, yeah, 3 minutes long. A lot of the other videos, honestly, were like 12 to 15 to 20 minutes long, trying to explain this. And so this one being 3 minutes long, I felt like it really stuck to kind of the basics. And then what I've done is I went through and I listed the steps for you while also telling you and kind of giving you the definition, if you will, of some of the enzymes that they talk about. And so as you watch the video, you might watch it once through, and then you might watch it and pause it as you start reading these steps. I think it'll make a lot more sense as you kind of go through it and then watch, and then go through a little bit more, and then watch again, so you can see these steps happening as they demonstrate them and describe them. Okay, uh, so again, it's only 3 minutes, but this could, if you're following my advice, it could take you 10 minutes to watch this and go through it. Okay, but it shouldn't really take too much longer than that. The goal, though, is that we understand these steps. So I'll go ahead and read the steps. The helicase is an enzyme responsible for unzipping the double helix. So again, in this video, you're going to see this helicase enzyme. It comes in and it unwinds the DNA, and then it starts separating those hydrogen bonds to create what's called a replication fork. And basically, you're just dividing, kind of like a zipper. As you unzip, you get to divide the two sides of your coat or your shirt. Uh, and so that's the replication fork. And again, you'll see that. So the helicase is an enzyme responsible for that. Once that happens, there needs to be what's called a primer added to the sequence of DNA. And so the primer is really like, hey, come and start at this point. Okay, start here, waving a big sign. And so the primase is the enzyme responsible for creating the starting sequence. So the primase then, once it's replicated, once the replication fork is created, the primase is going to attach to one of the strands, and it's going to lay down some RNA sequence for the primer. And really, I just want you to know the primase attaches to the single DNA strand and creates the primer. Okay, and this again is going to code and signal for the DNA polymerase. So it's really a signal, a starting signal to get the process started. So it tells the DNA polymerase where to begin. And that brings us to step 3. Once that primer, once that little waving sign has been attached to the DNA polymerase is an enzyme responsible for creating and attaching the new strands of DNA. So this really, the DNA polymerase is the enzyme that is responsible for copying and creating the DNA, the new strand of DNA, I should say. So the DNA polymerase attaches to the primer, then begins to place the correct DNA base pairs together as it moves along the sequence. And again, it's all in the video. I really encourage you watch it so that you can see kind of how it does that. Then the exonuclease removes all RNA primer sequences. So really, the DNA polymerase is going to keep on going, finishes the strand of DNA, which is a very, very long process. Relative, a lot of base pairs, but eventually it's going to be done. And so the exonuclease is responsible for removing the primer, because that start sequence actually kind of covers up some base pairs, and that's part of your DNA. But if it's in the form of RNA, then it will really kind of change your DNA and change your system. And if that happened every single time a cell replicated in multiple spots, that'd be a problem. So this new enzyme, the exonuclease, removes that little sign. Okay, picture you put a sign in your front yard saying start here. Okay, it removes that sign, the primer, and then the DNA polymerase actually comes back and goes, there's a spot missing, and then it fills in that little piece that it started with. Okay, and so anywhere that there are RNA primase, it kind of fills in those spots. Last but not least, picture, and maybe this is kind of like a race, right? A marathon. You have a start here, you have an end here, kind of sign. The DNA polymerase would be the runners. They go through the process. So then the DNA ligase is an enzyme responsible for cleaning up. Okay, you've got all this stuff. People were standing and watching. Okay, again with the race, there's going to be kind of different compounds and enzymes that might be kind of left over, and so the DNA ligase goes through and really cleans it up, and it puts the replication fork back together, which allows it to return to the shape. And it's going to do that for the 2 new copies, right? So you split it, and then you get copied twice. And so every time that happens, you're getting new copies that are completed. Okay, so again, that was a lot, but please watch the video, and hopefully that'll help us. But now you're going to practice basically being the DNA polymerase, and can you make the complementary sequence? So if the original sequence is AGGTCCG, which is right here on the right, okay, so that is the same sequence, can you match the left? Well, if I go back up here, and I know adenine and thymine go together, and guanine and cytosine. Okay, so I have adenine here on the right. This is the original sequence. Well, I know I need a thymine, so I can put a thymine there. G and C, G and A. Oop, well, that's not right, so I need a C up there. Okay, thymine and guanine. Well, that's not right. I need an adenine. Okay, so you can click and move these to create the matching sequence here on the left. Just like your DNA. It's DNA polymerase, sorry. Asking you to do that twice. Okay. And that really is the basics of DNA replication. And I think by understanding that, it'll then help us. Well, how then can the DNA be used to create proteins? And so, with this, again, I found another video, and this one, it's a little, I would say, a little more kiddish with the Amoeba sisters, but it honestly has really good visuals, and it breaks it down simply. And again, this one's 8 minutes long, and it is significantly shorter than all the other videos I could find, and it does a good job. So. Uh, I really encourage that you watch this video. But again, I tried to break down the process here. So with protein synthesis, there's two parts to it, and the first part is called transcription. There needs to be transcription of the DNA, and then the second part, and so we're going to learn what that process is. Ooh, and I want to make this bigger. The second part is going to be translation. Now that we've transcribed it, we need to make it readable for what's called tRNA to be able to attach the amino acids. So transcription and translation is really the foundation and the two processes that uh, create proteins. But now we need to understand them. So now that we know a little bit more about how DNA can copy itself and how the base pairs match and complement one another, uh, hopefully this will make some sense as well. So first, in the nucleus, RNA polymerase. So that, of course, that's different than DNA polymerase, but it's going to have a very similar function. The RNA polymerase is going to go through and create a copy. So just like when the DNA is made into the replication fork, and now it's able to make a copy of itself, the RNA polymerase is going to go through and create a complementary strand, but it's not going to be a DNA strand. It's going to be referred to as an RNA strand, and more specifically, we refer to it as a messenger RNA. So you will see it as mRNA most commonly. And the only difference, so you might be, well, what's the difference between RNA and DNA? One, we talked about this when we talked about carbohydrates and nucleic acids, but DNA is deoxyribose nucleic acid, and RNA is ribose nucleic acid. And so it's just the difference of an oxygen compound there in the carbohydrate. And then as far as the nitrogen bases, you're going to get a new base called uracil. And that is going to replace thymine. So you'll notice over here there is no thymine, but there is uracil instead. Cytosine, adenine, and guanine stay the same. But of course, that's a big difference for chemical standards, right? That's a whole new compound with different chemistry, and so it is completely different in the human body, and then is capable of different things. So uh, the DNA is going to create a complementary mRNA strand. That messenger RNA, that mRNA, then is able to do something that DNA is not allowed to do. It's not able to do. It is going to be carried out of the nucleus where it will find and attach to a ribosome. And so just a ribosome, all we really need to know, this is where the protein making is going to happen. So again, we took DNA in the nucleus, we made an mRNA copy, and we sent it out to a ribosome to start making protein. So I want to practice that. So again, similar to the DNA part, I want you to, can you make mRNA? So again, if you have adenine here on the right, what needs to go with it is uracil. So I would move a U up there with it. Just like that. And then there's still going to be thymine in the DNA. So what goes with thymine? Well, in the RNA, you have adenine still, right? So again, you can move these as you go through and get them to partner up properly. And again, here's the original sequence. So over here is the RNA sequence, the mRNA, I should say, and here's the DNA. I'm asking that you do it a second time again, just to get a feel for the difference, but hopefully if we understand it, it won't be too difficult. And then, quick question, where does the mRNA go after it is created? So after you've created that mRNA strand, where does it go? Okay, so that is the transcription part. You're transcribing the DNA into mRNA, and that allows it to leave the cell and then create these proteins. Or I apologize, it allows it to leave the nucleus to where the proteins can form outside of the cell. So now translation, and this is step 3. The ribosome now, with the mRNA inside of it, so you got this ribosome here as an enzyme that where the proteins are going to be made. The messenger RNA is the again, it's supposed to kind of look like the DNA strand, but it's messenger RNA, and it's only one half, right? So it has bonding sites that are open. Is going to go through this ribosome, and this will attract tRNA molecules that carry amino acids. And so scrolling down here real quick, tRNA is, we call it transfer RNA, is a type of RNA that is it's just floating around in the cytoplasm. It's floating around in the empty space of the cell. And if you look at this, right, tRNA, it has a small sequence of RNA. Okay, and again, this would be adenine, adenine, and guanine. So these are nitrogen bases. This is again just a graphic, right? But it's still going to have the carbohydrate. It's still going to have the phosphate, and now it actually has an amino acid attached to it, a protein. Again, amino acids are the foundations of protein, and multiple amino acids clumped together make a protein that can then do something. It can become something, right? So these tRNAs have a small sequence, and if this small sequence called a codon, that's going to be here. Okay, a codon has a sequence of 3. If it can find a match in the mRNA, AAG, UUC, then that tRNA can attach right here. It can bond. And when that bonding occurs, so you see here, these have bonded. When that bonding occurs, the amino acids will actually bond together. And once those bond together, that really triggers the tRNA to stop bonding, to let go, get out of here. And so that's where you see this tRNA really gets kicked out, but the amino acids are left bonded together. So again, this is where it starts, comes in, finds a sequence. Now, if this sequence doesn't match, it just kind of bounces off and goes back away. It's not going to bond here, or it's not supposed to, I should say. But then when you get these matches, that's going to tell which amino acids are together, and then it creates a growing peptide chain, a polypeptide, which again, those were terms that we talked about when we talked about proteins. So this is going to form proteins. So, and that's here in step 4. Once the tRNA is successfully attached to the mRNA, the amino acid will bond with the other amino acid in a sequence, forming a chain of amino acids called a polypeptide, which will form part or all of a protein. So the next part is, well, how does this kind of work? And here's what we know. Different codons, again, that's a grouping of 3 RNA bonding sites, if you will, or nitrogen bases. each codon will basically code for a specific amino acid. And so there's a list of amino acids. I'm not asking that you memorize them, but they're represented here. So PAG, for example, and I have it down here, PAG is coded for by a couple different things. You'll see that sometimes it can be coded by different codons. So UUU or UUC is going to create this phenylalanine. I think I said that right. Uh, and so that is something that we know and it can then predict. And so we can then do this process of creating these amino acids. So that's what I'm going to ask you to do here. You have to use this chart to try and identify the three amino acids that is going to be formed from the mRNA. So you have, for example, UCU. That is your first codon, right? There's three nitrogen bases there represented in that sequence. So UCU, I go through my chart. First letter is U, second letter is U, and then a U. So UCU is right there. I see that's SER. SER is serine. Whoops. So then I can drag that. Why does it not let me drag it? There it goes. I can drag that. That is the first amino acid. So now I want to look at the second grouping of three, ACU. ACU right there. THR. Yes, THR. So I can go down my list. Where's THR? There it is. Threonine. Don't know why it keeps doing that. Well, hopefully yours will work better. There it goes. And the third amino acid, GUU. And I see that right there. So then I track that up there. Valine. So those are the three amino acids that then will be bonded together. Now, right now, that doesn't mean a whole lot to us, but in short, to kind of summarize it, this is an introduction. But these three, they might be brown eye color. Those three bonded together in that order might signal and create brown eye color. Okay, it might signal, how long your hair grows. Okay, so these proteins again can really create everything, and they're going to be hundreds of proteins long, if not thousands or millions of proteins long, or sorry, amino acids long to form these proteins. So again, this is just the start of a potential protein here. And the order and the protein amino acids present make a huge difference. So now thinking about what I said, what happens if a mistake happens? So that's going to be the last couple questions here. I have this sequence, and we have the same sequence with one mistake. So there was a mistake that happened in the copying of the RNA and that mRNA sequence. And so I bolded that here for you. The only difference is that A used to be a C. Does that change the amino acids that are coded for? You have to try and figure that out. And then reflect on a hypothesis. What do you think that does? What happens? Is it good? Is it bad? Does it happen often? So I just want to kind of get an idea of what do you know? What do you think already? You don't need to research this answer, but this is where that conversation is going to go. All right. Thank you. Ooh.